Welcome back to regular lectures on CSIR-NET Life Sciences, and for those aspirants for NEET exams and the All Institute of Medical Sciences. Now we continue with the topic on biophysical methods and molecular biology, which is on Unit 13. We go ahead with the topic of immunological techniques, types, and samples. Now, most of the immunological techniques are based on antigen-antibody reactions. Precipitation reactions are one of the most important reactions that occur when antigen and antibody come to contact. When a soluble antigen reacts with its antibody in the presence of sodium chloride at optimal temperature and pH, the antigen-antibody complex forms an insoluble precipitate. Generally, liquid media and gels such as agar, agarose, polyacrylamide are used for this kind of reaction. This is a diagram which shows that the antigen binding site, variable constant, antigen binding site, immunological techniques, light chain, heavy chain. And we go on to the details of the techniques, immunodiffusion tests. Right, this is an immunological technique used to find out different antigens and antibodies in clinical examples. The tests are performed in 1% agar. There are some advantages of using immunodiffusion tests in a clinical setup, such as the band formed after the reaction is easily visible, stable, and can be stained for preservation. Different antigens can be used to observe the reaction, as each antigen-antibody reaction gives a specific precipitation line. Therefore, it helps to identify specific antigens. Identical, partial identical, and non-identical antigens can be observed. Single diffusion in one dimension Odin procedure. The antibody is mixed with agar in a test tube and the antigen solution is added over it. And as a result, antigen diffuses downward through agar gel and a line of precipitation is formed. The number of different precipitate bands will indicate different types of antigens. Double diffusion in one dimension, Oakley-Fulthorpe procedure. The antigen is mixed with agar in a test tube. A column of plain agar is added on top of the antibody solution. The antigen is poured on the plain agar column. The antigen and antibody moves towards each other through the intervening column of plain agar, and a precipitate band will form with the optimum concentration of the antigen and antibody. Single diffusion in two dimensions, radial immunodiffusion. The antibody is mixed with agar gel and a layer of this mixture is formed on a glass slide. Wells are cut on the surface of the gel. The antigen solution will be added to the wells. As a result, it diffuses and a ring-shaped precipitation band is formed. The diagram of the band is proportional to the concentration of the antigen. And this technique, the diameter of the band is proportional to the concentration of the antigen. This technique was used for the estimation of IgG, IgM, and IgA in sera, and for the screening of antibodies of influenza virus. Double diffusion in two dimension, Auster-Lani procedure. A layer of agar gel is formed on a Petri plate, and then wells are formed by using the template. Antibody solution is added to the central well and different antigens are added to the surrounding wells. If two antigens are identical, the precipitation lines will fuse. If two adjacent antigens are unrelated, the precipitation lines will cross. In the case of partially related antigens, spur formation will be observed. This technique is used for the toxicity test of C. diphtheria ELX test. Immunoelectrophoresis. Immunoelectrophoresis is a combination of electrophoresis and immune diffusion. A glass slide is used which is layered with semi-solid agar. A well will be formed on the surface of the agar and antigen solution will be added to the well. Electrophoresis will be performed for 1 hour. Then a rectangular trough will be cut parallel to the direction of migration of antigen. Antibody solution will be added to the trough. It will be left for 18 to 24 hours for diffusions. As a result, precipitation band will be formed based on each separated compound. This technique is used for the detection of different antigens in human serum and normal and abnormal serum proteins such as myeloma proteins. Counter immunoelectrophoresis. It is a one-dimensional double electrode immunodiffusion test. The test is based on the movement of antigen and antibody in the opposite direction. This test is also performed on a glass slide which will be layered with agar. 2 different wells will be formed on the surface of the agar. In one well, antigen will be added and another well will contain antibodies. The electricity will pass through this, which accelerates the movement of antigen and antibody towards each other. A precipitation line will be formed at a specific point between the 2 wells. This test is a sensitive standard technique and requires around 30 minutes to perform. This technique is usually for clinical detection of hepatitis B antigens and antibodies, antigens of cryptococcus in cerebrospinal fluid. Rocket electrophoresis. It is a one-dimensional single electroimmunodiffusion test. It is mostly used for the quantification of antigens. In this case, the antibody is mixed with the agarose gel, and this mixture will be used to form a layer on the glass slide. Wells will be formed on the surface of the gel and antigens added to those wells in increasing concentration. Electrophoresis will be performed. As a result, cone-like precipitation bands, rocket-like structures will be observed. The length of the rocket-like structures is directly connected with the concentration of antigens. Radio-immunoassay, RIA. This technique was first described by Persson and Yallo. It is mostly used for the quantities of hormones, drugs, hepatitis B surface antigen, IgE, and viral antigens. The test is based on the competition for a fixed amount of specific antibodies between a known radio-labeled antigen and an unknown test antigen. The competition is determined by the level of antigen present in the reacting system. At the end of the antigen-antibody reaction, the antigen will be found in free and bound fractions and their radioactivity will be measured. The concentration of test antigen is calculated from the ratio of bound and total antigen levels using a reference curve. Enzyme-linked immunosorbent assay, ELISA. The principal ELISA is based on an enzyme that acts on a specific substrate to produce a color. The color will indicate a positive result. Based on this principle, there are different types of ELISA, such as sandwich ELISA. Microtiters placed are used to perform this test. The wells of the plate are coated with a specific antibody against antigen to be detected. The clinical specimen is then added to the wells. If the antigen is present in the specimen, it binds with the coated antibody. This antigen-antibody reaction is detected by using antiserum conjugated with an enzyme. This antibody attaches to the antigen which is already bound with its specific antibody. A substrate is then added to the reaction mixture which will bind with the conjugated enzyme. In case of a positive result, the enzyme substrate complex will produce color, and intensity of the color is then further read by the ELISA reader. Positive and negative control should be performed along with the test. Indirect ELISA. This test is used for the detection of antibodies. The wells of the microtiter plate are coated with antigen. A clinical sample of sera is added to these wells. If the specific antibody is present in the sample, it will form a complex with the antigen coated in the wells. This antigen-antibody complex is detected by adding enzyme conjugated immunoglobulin, which will bind with the antibodies of the specimen. A substrate is then added, which forms a complex with the enzyme and produces color as a positive result. Positive and negative controls are equally important to perform this test. The enzymes which are generally used are horseradish, peroxidase, alkaline, phosphatase, etc. The substrate used for horseradish peroxidase is O-phenyl diamine dihydrochloride, and the substrate for alkaline phosphatase is p-nitrophenyl phosphate. Competitive ELISA. It is used for the detection of HIV antibodies. It is different than the previous two types of ELISA. In this case, the appearance of color indicates a negative test, while no other color change indicates positive results. In this case, the competition occurs between an enzyme-linked antibody and a test antibody that is present in the clinical sample. These two antibodies compete for the same antigen. The wells of the microtiter plate are coated with the antigen of HIV. The sera test sample is added to these wells and incubated at room temperature and then washed. If the specific antibody is present in the test sample, the antigen-antibody reaction will occur. Enzyme-labeled antibodies are then added to the reaction mixture to detect the antigen-antibody complex. The plate is further washed, and after incubation, if the test sample contains the antibodies of HIV, then these enzyme-linked antibodies will not be able to form a complex with the antigen and will leave the reaction mixture after washing. Then the substrate is added, and as there are no enzyme-linked antibodies present in the system, no color change will be observed as a positive test. Uses of ELISA: detection of HIV antibodies in serum, detection of mycobacterial antibodies, detection of rotavirus in feces, detection of hepatitis B markers in serum, detection of enterotoxin of E. coli in feces. The next procedure will be single diffusion in one dimension Odin procedure. The antibody is mixed with agar in a test tube and the antigen solution is added over it. As a result, antigen diffuses downward through agar gel and a line of precipitation is formed. The number of different precipitate bands will indicate different types of antigens. Double diffusion in one dimension Oakley full stop procedure. The antibody is mixed with agar in a test tube. A column of plain agar is added on top of the antibody solution. Antigen is poured over the plain agar column. It's just a repetition, and we'll stop it there. The antigen and antibody move towards each other through the intervening column of plain agar, and a precipitate band will form when at optimum concentration of the antigen antibody. The next is an interesting science video for trash and environment. Just for the, it's has a lot of info to be conveyed. Please watch. Well, there's a pause in the video. We'll watch it later. Immunodiffusions test. This is an immunological technique used to find out different antigens and antibodies in clinical samples. The tests are performed in 1% agar. There are some advantages of using immunodiffusion tests in a clinical setup, such as the band formed after the reaction is easily visible, stable, and can be stained for preservation. Different antigens can be used to observe the reaction, as each antigen-antibody reaction gives a specific precipitation line. Therefore, it helps to identify specific antigens. Identical, partial identical, and non-identical antigens can be observed. Single diffusion in one dimension Odin procedure. Antigen is mixed with the agar in a test tube, and the antigen solution is added over it. As a result, antigen diffuses downward through agar gel and a line of precipitation is formed. Number of precipitation bands will indicate different types of antigens. And some Q&A on this topic. What are the immunotechniques? Immunotechniques are methods used to study the immune. Immunotechniques are methods used to study the immune system and procedure of antibodies that can detect specific proteins in biological samples. They are used for inducing, measuring, and characterizing immune responses. To keep you informed. Right, the topic would be immunotechniques. Immunotechniques are methods to study immune system and to production and use of antibodies to detect specific proteins in biological samples. Uses of immunotechniques. Immunotechniques are used for inducing, measuring, and characterizing immune responses. These can alter immune systems through cellular, molecular, and genetic manipulation. And production of antibodies. Antibodies are produced from cells or animals in sufficient quantity to be used as experimental reagent. Monoclonal antibodies are produced by injecting mice, and polyclonal antibodies are produced by injecting rabbits or goats with live or inactivated material and collect the blood of those animals that have high titers to the target. Antibody isolation and purification. Excuse me. Antibody isolation and purification is to keep specific antibodies and removal of non-specific antibodies. Pure isolated antibodies have greater activity and are less likely to cross-react with non-target molecules. Antigen-antibody reaction. Antibodies are produced by the immune system of vertebrates and are essential for prevention and resolution of infection. An individual antibody can react with only one antigen. Antibodies are glycoproteins that bind specifically to antigen. The interaction between an antigen and antibody depends on non-covalent bonds. Low affinity antibody and bind antigen weakly and tend to dissociate, whereas high affinity antibody bind antigen tightly and remain bound longer. Precipitation reaction. It occurs in fluids. Reaction of soluble antibody in aqueous solution form a lattice insoluble Ag silver AB complex that within 1 minute that develops into a visible precipitation in 1 to 2 days. AB that aggregate soluble Ag are called precipitants. Formation of insoluble AB-Ag complex occur in a concentration range known as a zone of equivalence. Immunodiffusion tests. When Ag and Ab diffuse towards one another in agar, or when Ab is incorporated into agar and Ag diffuses into the Ab containing matrix, a visible line is formed. Indirect agglutination test, indirect or passive agglutination test, and use of agglutination tests. Direct agglutination. When the antigen is an integral part of the surface of a cell, it is called direct agglutination. Indirect agglutination, when coated cells are used to, it is called indirect or passive agglutination. Hemagglutination. Clumping of RBC is called hemagglutination. Clumping. Immunoassays. Immunoassays are bioanalytical methods that identify and quantify analytes in biological samples. Immunoassays are based on the specific ABAG antigen reaction. And competitive immunoassays label and unlabeled antigens exposed to the antibody. In non-competitive immunoassays, the labeled antibody detects the bound antigens. It is of two types. Radioimmunoassays, RIA. RIA is a technique for determining the concentration of a particular antigen in a sample based on competitive binding between non-labeled and labeled antigens for a specific antibody. Basin yellow developed at the RIA method. It is most sensitive technique for detecting Ag or Ab. Enzyme-linked immunoabsorbance assay, ELISA, is a sensitive method to quantify or test the presence of an antigen. Enzyme-linked AB are used to bind and light on a solid substrate and convert a colorless molecule to a colored product for detection. Immunofluorescence, AB could be labeled with molecules that have the property of fluorescence. Fluorescent molecules absorb light of one wavelength and emit light of another wavelength. The most commonly used fluorescent dyes are fluorescein and rhodamine. Immunoblotting. Immunoblotting is an in vitro technique of detecting specific proteins using antibodies. Protein mixtures are transferred to a filter membrane either directly or after separation by electrophoresis, then probed with antibodies that are either labeled by fluorophore or an enzyme or bound by a labeled secondary antibody for detection. It is also called Western blotting. Immunoelectrophoresis. The AG mixture is first electrophoresized to separate its component by charge. Drops are then cut into agar's gel parallel to the direction of electric field. Antiserum is added. AG and AB then diffuse towards each other and produce lines of precipitation. It is used to detect the presence of proteins in serum. This diagram depicts that. And then in immunohistochemistry, a technique used for detecting molecules of interest within tissues using antibodies, probes, or enzymes conjugated to primary antibodies that bind the target molecule secondary antibodies. So that's it about that. We now have come to the next topic, immunochemical techniques. Introduction. All vertebrates have advanced immune system. The more complex the organism, the more advanced the immune system. Right? And now the immune system of mammals has evolved over 1 million years. It provides an incredible protection system capable of responding to infective challenges that arise in the body. Immunity in our body is monitored or controlled by specific cells, from stem cells and bone marrow. The most important cell types are B and T lymphocytes, which have the ability to act against bacterial and other virus. B cells release antibody against assimilation of foreign substance in the body. An antigen is a foreign substance capable of an immune response leading to the production of antibodies. Remember, antigen is always a foreign body, which produces antibodies within the body of the host. Okay, they are the targets to which antibodies bind. So antibodies are antigen specific, bind to the antigen that initiated its production. This unit focuses on the identification and diagnostic chemical techniques on the response of an antibody to a specific antigen. Immunochemical methods are based on selective, reversible, and non-covalent binding of antigens by antibodies. These methods are employed to detect or quantify either antigens or antibodies. Objectives. After reading this lesson, you will be able to define immunochemical techniques, explain the characteristic features and roles of antigen-antibody reaction, describe and distinguish the type of immunochemical techniques. Immunochemical techniques, factors controlling the immunochemical techniques. All the techniques cannot be used for identification of a specific antigen or antibody. In order to perform the immunochemical techniques, several controlling criteria are performed. Experimental conditions, nature and the place of work, type of sample collected, nature of reagents, the quality is studied, standardized, and analyzed, sensitivity and selectivity of technique to the particular sample. Immunochemical techniques. Immunochemistry is an advanced area of immunology. It deals with the chemical components and chemistry, that's chemical reactions of immunological phenomena, that is of antigen-antibody immunochemical methods and processes utilizing the highly specific affinity of an antibody for its antigen. It detects the distribution of a given protein or antigen in tissue or cells. The methods used for the immunochemical analysis are called immunochemical techniques. They are highly important in diagnostic and clinical context, as now even normal cell with many proteins are altered in disease state in cancer. Characteristics and role. Characteristics. Simple, rapid, and robust, highly sensitive, easily automated, applicable to regular clinical diagnostic laboratories, does not require extensive and easily destructible sample preparation, do not require expensive instrumentation, mostly based on simple photo, fluoro, and immunolumiноmetric detection. Measurements may be either qualitative or quantitative. Roles. The characteristic features of these techniques are helpful for the following. Function of any newly identified or novel proteins can be identified. Imports of uncharacterized protein in the natural environment can be analyzed. Next would be continuation of the immunological techniques, immunochemical techniques. Determination of species of tissues in which the protein or residue is expressed, the cell type or subcellular compartment in which the protein can be found, detect whether there are any variation in expression of protein during development of the organism. Some alteration in the normal expression pattern of a particular protein may indicate a particular disease state, gives information that may be useful for diagnosis and treatment methods of analysis. All immunochemical methods are based on a highly specific and sensitive reaction between antigen and an antibody. Antibodies are immunoglobulins belonging to the family of glycoproteins IgG, IgA, IgD, IgM, and IgE. Structurally, antibodies are often visualized as Y-shaped molecules, each containing 4 polypeptides, 2 identical polypeptides, unit called heavy chains and another 2 called light chains. It has a domain called Fab, the site where it binds to an antigen. The region of an antigen binding to an antibody is called the epitope. Take this screenshot and make notes, please. The measurement of the strength of the binding is called affinity, and it is usually expressed in terms of the concentration of antigen-antibody complex measured at equilibrium. It is measured by quantitative precipitin curve, basis for many immunochemical tests, proposed by Heidelberg and Kendall in 1935. Immunochemical techniques. Quantitative precipitin curve. It describes the relationship between the antigen concentration and the amount of precipitate for a constant quantity of an antibody. 3 zones can be distinguished from the precipitin curve. Please note this. Take a screenshot and take these notes too. Antibody excess zone, first phase where less antigen is present in sample. Equivalent zone, both antigen and antibody are cross-linked, forming precipitate, no free antigen, no antibody is present. Antigen excess zone, amount of precipitate reduces due to high antigen concentration. The precipitin curve forms the basis of most of the immunochemical techniques that can be performed in clinical laboratories. Now the text questions. 24.1. Dash cells release antibodies against stimulation of a foreign substance in the body. It's an antigen antibody, right? So antigen, when an antigen comes, you have an antibody cells. So antigen cells release antibody against. Let's fill up the answers. Dash is a foreign substance capable of inducing the production of antibodies. Antigen. Dash methods detect or qualify either antigen or antibodies. The measurement of the strength of the binding is called. And your measurement of the binding, so what is it? I guess you'll find out the answers and then put them in the comment section. I will get back to you, right? Types of antibody used. The antibodies used for the methods are produced in various ways. Monoclonal antibodies, products of a single clone of plasma cells by B lymphocytes, mostly prepared in laboratory. They are directed against single epitope, identical copies with the same structure and antigen specificity. They have excellent specificity but poor ability to precipitate antigen. Polyclonal antibody. They are conventional, that is produced by immunization of animals with antigen. The antibody consists of a mixture of monoclonal antibodies having specificity for complex antigens. Sometimes monoclonal is called as monovalent and polyclonal as polyvalent, which indicates antigen specificity. Types. Based on the type of reaction performed, reagents and samples used, the techniques are categorized as follows. Particle methods, where the antigen-antibody interaction is observed. It includes agglutination, immunoprecipitation, immunoelectrophoresis, immunofixation, immunoturbidimetry, immunonephilometry. Label methods. If the antigen antibody is labeled and through the label concentration the antigen-antibody reaction is observed, this includes immunoassay, competitive binding. Other methods include immunofluorescence, immunoelectron microscopy, etc. Agglutination is the next term. Agglutination from Latin as agglutino, to gel or to glue or attach, is a process of formation of clumping of cells. It occurs due to reaction of antibody or on a particular antigen. Among all of the antibodies, IgM is a good agglutinin since it has a high affinity to different antigens. There may be either a qualitative test to identify the presence of an antigen or an antibody, qualitative test to measure the level of antibodies to particulate antigens, serial dilutions of sample is used. There are many variations in agglutination methods. Direct antigen present on cell surface is directly agglutinated by specific antibodies. Example, in hematology, determination of blood group ABO typing, diagnosis of salmonella typhi, and Widal test. Indirect reverse agglutination uses particles already coated with antigens antibodies to determine the antibody antigen in given sample. Example, pregnancy test using hormone secretion, human chorionic gonadotrophin, diagnosis of rheumatoid arthritis, agglutination inhibition, where competitive binding of antigen occurs, determine the concentration of soluble antigen in sample. Hemoagglutination involves reactions using red chemicals, detection of diseases and other viruses, blood typing, etc. In-text questions. 24.2. The types of antibody used are dash and dash. Dash is the process of formation of clumping cells. Example of direct agglutination is dash typing. Example of indirect agglutination is dash test. Please find the answers and put them on the comment section. I'll grade your answers, all right? Immunoprecipitation. There are methods to include flocculation and precipitation reactions. When a solution of an antigen is mixed with its corresponding antibody under suitable conditions, the reactants form flocculating or precipitating aggregates. They can be assessed visually by the formation of precipitin line at the region of equivalence. Where equivalent amount of the antigen and antibody are present, it may either be simple reaction of one antigen and a precipitin line body, or complex when many unrelated reactants are used. Watch this diagram, take a copy, screenshot and study on it. And simple methods, a concentration gradient is established between the antigen and antibody. It includes single radial immune diffusion, SRID, developed by Mancini, simple technique where the circular precipitin area originating from antigen well in gel is equal to amount of antigen concentration. Double immune diffusion, developed by Orgeloni, both antigen and antibody diffuse from separate wells in a gel to form a precipitin line. In complex methods, different antigens are compared with an antibody and vice versa. The result is based on the presence or absence of precipitin line. Immunoelectrophoresis is a qualitative technique. It combines two methods, one after another. Gel electrophoresis, separation of components by charge, and immunodiffusion, diffusion of both antibody and antigen toward each other, produces precipitin line. Some figures, please take screenshots. It is strictly qualitatively technique to detect antibody and antigen samples, but also a quantitative technique exists called rocket immunoelectrophoresis to measure the antigen's level in the sample. Another variation called as counter current immunoelectrophoresis, where it is similar to double immunodiffusion. Immunofixation. Immunofixation is used for detection and identifying antibody in serum, urine, and other body fluids. The principle is similar to immunoelectrophoresis, involves two stages. Electrophoresis, separation of antibody proteins. Immunoprecipitation, separation with specific antibodies. It is easy to perform, more sensitive and easily evaluated. In clinical laboratories, is used for detection for cancerous myeloma to specific antibody. This is immunofixation serum in urine samples. And here, immunoturbidimetry, the precipitation line or curve that forms in a gel can also be formed in a solution, or when an antigen solution is combined with a specific antibody solution. The clumping formed results in a cloudy appearance. This is called turbidity. It forms the principles of immunoturbidimetry. It is measured based on the intensity amount per amount of light that passes through the sample. It is performed by an instrument called spectrophotometer. It's a diagram. Please take a screenshot. Immunonephelometry. Immunonephelometry works on the principle similar to turbidimetry, but this method measures the intensity of scatter light from sample directly. It uses laser light and is measured by using a nephelometer. Now, both the methods are faster but are more expensive, increased sensitivity of antigen-antibody can be got by automation of these techniques. Immunoassay. Immunoassay technique works on the principle of labeling either the antigen or antibody before the reaction. This increases the sensitivity of detection in the experiment and the formation of immunoprecipitate. The label can be of a radioisotope called a radioimmunoassay enzyme reaction known as enzyme immunoassay EIA. It also includes a ELISA enzyme linked immunosorbent assay, fluorescent substance, chemiluminescent substance, RIA, first immunoassay technique based on the measurement of radioactivity associated with antigen-antibody complexes. And ELISA, safer and efficient method based on the measurement of an enzyme action associated with antigen-antibody complexes, most commonly used enzymes, biotin, alkaline phosphatase, etc. This diagram is important. Take a screenshot. Solid phase, tetrabiodin, biotin-related antibody, antigen, and label antibody. Competitive binding. Another important to measure the amounts of antigen is competitive binding. In this technique, it works on the principle of competitive binding. Antibody is the first incubator in solution having antigen. This mixture is added to antigen-coded reaction. Well, it is the more the antigen present in sample, the less antibody will bind to the coded antigen. To this, when labeled antibody is added, we can measure the amount of antigen present in sample. Another method, immunofluorescence. Antibody label with a fluorescent molecule, fluorescein or rhodamine, or one of many other fluorescent dyes is used to detect the presence of an antigen in or on a cell or tissue by the fluorescence emitted by the bound antibody. Immunoelectron microscopy uses the specificity of antibody to view tissues and its components through a microscope. Immunostaining, use of antibody labeled with enzyme or dye to detect specific protein in a sample. Most important staining method is Western blot method that includes three processes: electrophoretic separation of proteins in sample, immunoblotting transfer protein from gel to a membrane, immunodetection identification using labeled antibody. Detection. In order to detect and diagnose disease and alter certain body conditions properly, there are many fast immunochemical techniques. Some examples are detection of salmonella typhi using typhoid in blood, detection of narcotics in urine and saliva, detection of protein hormones like insulin, thyroxine, etc. Pregnancy test for level of human gonadotrophic hormone, HCGH. What have you learned? Immunity in our body is monitored or controlled by specific cells from stem cells in bone marrow. The more important cell types are B and T lymphocytes, which have the ability to act against bacterial and other viruses. B cells release antibody against stimulation of a foreign substance into the body. An antigen is a foreign substance capable of an immune response leading to production of antibodies. Immunochemical methods are based on the selective, reversible, and non-covalent binding of antigens by antibodies. The methods used for immunochemical analysis are called immunochemical techniques. All immunochemical methods are based on a highly specific and systematic reaction between an antigen and an antibody. Antibodies are immunoglobulins belonging to a family of glycoproteins IgG, IgA, IgD, IgM, and IgE. The measure of the strength of the binding is called affinity. Quantitative precipitation curve describes the relationship between the antigen concentration and the amount of precipitate for a constant quantity of an antibody. Types of antibodies used in monoclonal antibodies, polyclonal antibody, they're the ones used. Based on the type of reaction performed, reagents and samples used, the techniques are categorized as follows. Particle methods where the antigen-antibody interaction is observed includes agglutination, immunoprecipitation, immunoelectrophoresis, immunification, immunoturbidimetry, immunonephelopmetry. Label methods is either antigen or antibody is labeled through the label concentration. The antigen-antibody reaction is observed. This includes immunoassay, competitive binding. Other methods includes immunofluorescence, immunoelectron microscopy, etc. Terminal questions. Give an account of characteristics of immunochemical techniques. What are the different methods of analysis? Please post them on the comment section. We will get back to you. Other questions are: discuss the various methods of agglutination and enumerate the differences between electroturbidimetry, immunoturbidimetry, immunonephelopmetry. Compare immunoprecipitation and immunoelectrophoresis. Give the importance of immunoassay. Mention its types. Answers to in-text questions are here. Okay, that's it about that chapter.